präsentiert von OpenLearnWare, die Plattform für Lernmaterialien an der TU Darmstadt.
It's a pleasure for me to be here. You're a bit of a new audience for me. I normally do not speak to people in your area. That's precisely the reason why I am pleased to be here, because the message that I carry, I think, has applications outside of the health sciences, which is the region in which I'm most comfortable with. So I would like to go into a discussion whereby we describe some recent findings in how the central nervous system, that is the brain and the spinal cord, control the generation and the modulation of a force in a muscle and to some extent in a group of muscles. For precisely the reason that Andre mentioned, there are lessons to be learned there, but I want to add also that there are precautions to be learned there. And I would focus probably a bit on the precautions by the end of the lecture. The human body is not a robot. It is not built like a robot. Therefore, it should not surprise us too much if it is not controlled the way a robot ought to be controlled. So keep that thought in mind as I go through this discussion here. I will focus mostly on how the muscles in the human body are controlled, but from time to time I'll try to draw inferences to what is good and what is less interesting about them vis-a-vis the notion of controlling robots. If along the way, I'm going to need some water. If along the way some of what I say is not clear to you, please stop me. Ask me questions. I don't mind at all. It's much more important that you understand what I'm trying to say than that you pretend you understand what I'm trying to say. Because then you're wasting your time and I have 7 hours jet lag for no good reason. Okay? So, be free. So, I don't know if this is familiar or not familiar to you, but I thought I would start with this slide to put us into a framework of what I'm going to be talking about. This is a muscle. These are groups of muscle fibers inside the muscle, grouped together in terms of motor units. That is, each of these groups here, or set of muscle fibers, is innervated by a branch of one single motor neuron that has its cell body in the spinal cord. So if I cut you like this, and I look down like that, in the front part of the spinal cord is where these motor neurons have their cell body. And this is one, and this is number n, and you have many of these different numbers for different muscles. And what happens is, if I activate a neuron that sits in the central sulcus in the middle part of the brain to say, I want to make a contraction. Magically, this happens. We do not know how that happens, but it happens. Pulses travel down here to the anterior horn cell. There's a synapse or a relay to the alpha motor neuron that go to this muscle fiber here, and the muscle fiber contract. The rate at which these pulses arrive, they contract faster and faster and faster. And eventually, they contract too fast for them to be individually identified, and you tetanize, that is, you fix the muscle and generates more or less smooth force. And if you do that, you see action potentials, electrical pulses, that travel along these muscle fibers. When you add all these up, you generate what is known as an EMG signal. Are you familiar with electromyographic signals, EMG signals? Is this a new concept to you, or are you more or less familiar with it? Please, don't be shy. Those of you who are familiar with it, please put your hands up. So, good point. All right. Good. Now, what of course a muscle is designed to do is not to generate an electrical signal. A muscle is designed to generate a force. And what we really would like to have done here is to on this pink stuff. That is, every time this twitches or contracts, a force pulse is generated. That is known as a force twitch. It's just a pulse. And every time it fires, it goes bang, bang, bang. And the smaller ones, which are the earlier ones to be recruited, fire faster. We'll see that, and they tend to superimpose. This is what produces force in the muscle, but we can't study that because you can't go inside the muscle and measure the force twitches of the individual muscle fibers. It's technically not feasible now. What we can do, though, is we can study these electrical pulses, because relatively speaking, they're much more easy to obtain. And we can look at their characteristics and try to understand what the heck really goes on inside the nervous system as a consequence of that. Okay, so I'm going to focus the rest of the lecture at trying to understand what's going on in these blue lines here, which are the action potentials, which is the term given for the electrical pulse that travels around the nerve and the muscle fiber. All right? By learning that, we learn something about how these force twitches are controlled and something about how this force is controlled. So, there's an action potential traveling down at the muscle fiber, it reaches there, and this slide is not working. It's supposed to go twitch, 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 twitch. Something happened along the way. But basically what happens is the action potentials fire, bang, 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 bang. Every time one of these occurs, a force twitch occurs in the muscle, and when these fire faster and faster, as you see here, they add upon each other to a point where if you were to fire them even further, no force is generated. So there's a limiting factor inside the system, something to remember for later. So, I'm going to give you now a poetic schematic. This is not exactly correct, but it's visually simplified to give you the point of how the motor units are controlled in the muscle. So here we are at the anterior horn cell, that is, I've cut him up, remember? I'm looking down in the spinal cord, and this is the face that I see. Coming from there will be a motor unit, M1 on the previous slides, which is a group of muscle fibers, all innervated by one nerve. And there's some noise in the system. There's a lot of noise in the neuromuscular system. And that's the first one that becomes excited, then the next one, and the nth one afterwards. You notice that the first one has a small force twitch amplitude, and they're progressively larger and larger until the very last one has the largest of all. So watch what happens. That one's become excited, starting to fire, generating force. And this one here gets excited, and that one starts generating force. So we will, much of what I'll talk about for the next 15 minutes are the details of this slide here. But here it started firing, it got faster and faster, and it built up the force from this force twitch. This motor unit 1 to motor unit N. If you add all of those up, this, by the way, is simply a convolution of this firing rate with that twitch. You get this force. The question is, how does this work? How are all these motor units organized? First of all, are they organized? Secondly, how are they organized? And thirdly, how are we being taught? I'm sure those of you who have taken courses in simple physiology will have been taught something different than what I'm going to talk about. And we will discuss that as to why I believe it's different. But there's still much unknown about this. I said this one started first. Well, probably. I said this one started later on in this last one, and I made some assumption about the firing rate, that these are slower than these ones here, and so forth and so on. Now let's go into the detail of how correct that is. And if I were to repeat that without the force, instead of the first, the action potentials, to look to look at, instead of the force twitches, to look at the action potentials of each motor unit, I would get this structure here that gives me that EMG signal that we talked about. And again, I'm focusing on this because this we can study. We can measure this. We can measure that. We can measure this. We can measure this, this, and this. So let's go further into this. Now, how do you do that? And this has been the problem for the past 60 years. Because to look at the firings of many motor units at the same time when they are activated, you end up with a very complicated looking EMG signal, like the superposition structure that I gave you before. And it is very difficult to go into that signal and look at the individual firings of each motor unit. So what has been done historically is either they have put needle sensor inside the muscle and looked at just a few. Very difficult to do. I want you to do that experiment sometime. You get a needle shoved into you, and it says, please contract hard. It's not easy. So what we have done is we have developed a technology over the past 30 years that recently in the past 7 or 8 years has really blossomed. And that is, we put a surface sensor on the skin above the muscle that we want to study. We collect an EMG signal, then we decompose it through algorithms that have been evolving for decades, literally, so that we can decompose this complex signal into the individual firings, each individual firing of each motor unit, whose electrical activity is detected by this sensor on the surface. This is a significant feat. We have developed technology to do that, and the sensor is right here. It's not a very good picture, is it? I'm sorry, I don't know what happened to my slides. But anyway, this is the sensor. It has 5 small prongs that come out, which are detectors on the surface of the muscle. And through amplifiers and through the decomposition algorithm, you get the data that we're looking for. How that works is it records 4 channels. This is the sensor. It has 4 prongs that stick out. It records 4 differential channels, and this is the type of signal that then is passed through the algorithms to be decomposed, and we're going to take this signal and identify within it, as I said before, every discharge, or 95% at least, of each of the discharges for every motor unit, and we'll know how many motor units also. Okay? These are just the real data that you have to deal with in order to accomplish this. If you do that, then you take, here are the 4 channels of the signal again. This is a blow-up of the signal to show you the complexity that exists in the signal. If you then decompose these signals in 4 channels, you get, in this case, we identified 30 simultaneously activated motor units, each having a unique shape. And this is the firing. Each bar is the time at which this electrical pulse occurs for that specific motor unit. This decomposition showing the timing of the firings is accurate to about 95.4% on the average, which is very good for this kind of work compared to alternative techniques which are unable to see this number of motor units at a time and nowhere near that accuracy that I have just mentioned. So I'm not going to dwell on the technology. Take it as fact. This has been highly criticized in a positive way and has been proven independently to be correct by other researchers. Here's an example of how the decomposition would work. This is one of those signals I showed you before. You go through the decomposition process. This is the force that the muscle generated to provide this EMG signal here. And you're going to see the first motor unit, the firings appear here. And at this point here, we're going to see the firing rate. That is the averaged inverse of the number of firing pulses per second. So this filter here is really a Hamming filter, which provides the firing rate for each motor unit. Watch here and watch something go up right there. So here's the first motor unit, and there's the firing rate. It happens very quickly, but you'll get a chance to see for it. That's the first one that was identified. The last one, which is identified, will appear up here, and the firing rate will appear somewhere down here. There it is. So this is number 1, and this is number 30 of what I showed you in the previous slide. So this one is firing fast, so here's the firing rate. This is pulses per second, and this is the excitation time, the excitation, that is the degree of the force. MVC is maximum voluntary contraction. That's the maximum force you can produce from that muscle. So this is 0 force, this is maximal force, so this is 50% of maximal force at this point. And then in between, you have all the other motor units. This is part of the diagram that I showed you in the previous slide. That's the first part of it. But did you notice something rather interesting here? The first motor unit that was found was the smallest amplitude, fired the fastest. That's the green line. The last one had among the largest amplitude, but fired the slowest. That was in here. And in between, there is a hierarchical, progressive change in the firing rates of motor units. This system is highly structured. The motor units respond uniquely to an excitation, but all structured in a progressive way. The first one that gets recruited, that performs the smallest level of force, fires the fastest. The latter one, which provides much higher force impulse, fires the slowest. Now, if you have taken courses in physiology, you will recognize this as being precisely the opposite of what is currently taught and has been taught for a while. Why? The work of Eccles in '54 was the dominant work in this area. They cannot do this, right? Remember I told you there's a technology issue until recently. What they did is they electrically stimulated the nerves that went to a muscle by taking cats, decerebrating them, that is cutting the brain off from the spinal cord, then dissecting the area where the nerve went into the muscle, put some wires on the nerve, electrically stimulated the wires, and watched what happened in the muscle. And they did that one at a time. And what they saw was that the higher threshold motor units, the ones that fire at the last, fired the fastest. It's not what this says. It said fired the fastest. Yes?
You said that the last motor units had the highest amplitude.
And also the highest force twitch. Yeah, it's a very, very complicated algorithm. It's a set of algorithms that has about 250,000 lines of code in it. It's a serious artificial intelligence issue that we need to discuss. And I will be happy to answer that question, and to anybody else who's interested in this question after the talk or whenever there's time. It will take about 15 to 20 minutes to explain that. It's a very good question. It's an important question. And I'm asking you to accept it on faith right now, but I will give you a description that will hopefully satisfy you, and I will also reference you to various published works that describe this in great detail. It's a great challenge. It's one that took decades to overcome. Is that sufficient for now? Okay. Please, I'll be happy. This is what I like. To me, this is the good stuff. Okay. Now, But what this says is the following, that when an activation, when the central nervous system, through the spinal cord, provides an excitation, it goes through the muscle, and the muscle responds like this, all the different motor units in a muscle. This is, I've shown you from one muscle here, but we've done this for many muscles. The characteristics change, the structure does not. So the muscle responds in unison, but the motor units within the muscle respond with their own particular characteristics, which are hierarchically structured. So once you have an input to a muscle, the output is determined by the hierarchical structure of the characteristics of the muscle fibers within that muscle. You can think of it as a simple input-output system that has a structural behavior inside. But so it could be a simple linear system from input to output, where the internal structure, however, is highly organized, but always responds the same way. Now, and just to show you, this is another example where we have a force. This is the force that we generated from the muscle, and these are all those fine rates which which we have been able to decompose as we did in the previous slide. But when we don't filter them very much, there's only a 0.5 second Hamming filter here. You get this very complicated looking structure. What happens when you increase the time of the Hamming filter to 1 second? You notice how it starts to clear up? We're removing some of the noise, and now we increase it to 5 seconds, much easier, much smoother. We filter it to 10-second width for the Hamming filter, and look at what you see. As you remove the noise which is in the system, this is what the brain is actually sending. This is the signal that this is how the motor units respond. The first order response to what the brain is sending to the muscle fiber. Beautifully structured. The first ones begin firing at a faster rate and continuously have a faster rate. The last one begin firing at a lower rate and continuously have a lower rate. Now, speed up. We can, oops, sorry. We can, and I'm not going to go into this, and there's questions that should be answered at this point here, but I want to get to the major issues. We can generate equations. These are the behavior-defined rates. We can do some curve fittings, and you can see that these are all one minus exponential kind of curve fittings for different force levels at 100%, 80%. Anyway, we can generate the equations for the defined rate as a function of time, as a function of force, and as a function of recruitment threshold, which is where they begin to study. This becomes a very long, complicated equation, which can be solved, and it looks I just explained this. Looks like that. We can actually formulate the response, the fine rate response of a whole muscle. With these, and these are the equations that come, this is the equation that's solved from that synthesized equation I put before. And now you can think of it the following way. You have mathematical equations that describe the fine rate versus excitation. The first one starts at a higher level and continuously goes to a higher level. This one at a lower level, continues at a lower level. If you draw a point somewhere along this excitation, say you want to perform a 50%, a 40% contraction, MVC level, you would define with a line here, this would be the operating point of the muscle. And the fine rates would be, for each motor unit, would be the values that intersect this line along the way. So you can now think of a muscle as actually is a set of equations. They are structured. And the whole muscle can be defined to have an operating point at the level at which it needs to contract. And the mathematical equation provides you the response of all the motor units. And you can move that up and down. What is important about this, is that here's the important one. At any excitation level, at any force level, and at any time, earlier recruited motor units have greater flying rates than later recruited motor units. Pick any point on here, and that is true. Or the flying rate of a motor unit is inversely proportional to the recruitment threshold. The ones that start highest, and so forth. This is all you need to remember if you're trying to design a controller for the muscle, the way the muscle is built. Now, this muscle is not a motor. It is not an activator. It has its own peculiar physiological aspects that it needs to have for many other reasons. So the control is structured in this way. One of the things I want to try and dig out in the next 15, 20 minutes is that may not be the case of how you really want to design a robot, though, because you don't have to deal with muscles the way they are built. The central nervous system has to deal with muscles the way they are built. Now, what it's done, it tried to simplify things for itself. It says the motor units are in happenstance. They don't fire as they wish when they want, and so forth and so on. There is a simple structure to them all. So the central nervous system sees this. It says, I want to contract at 40%. And so it sends excitation that cuts these flying rates at that point, and these things come out of the muscle. And that happens. If it's at 20%, this line will draw here, and that happens. My perspective is that this is what the brain sees. When we go down to the muscle, we get these very, very, very complicated views, because what we get is the information that the muscles provide us as they, the motor units, individually have to respond to what the brain is telling them to do. It looks very complicated downstream, but upstairs, it's probably something like this. The brain knows that if it does this, the muscle will produce X amount of force. How it happens is through this organized structure, but that's something that it's an terminal device, the muscle. So if we were to activate at this level here, all the motor units to the left would be active, and these ones, the gray ones, will not yet be activated because they are not needed to produce this level of force. Okay? That's what that means. And as you move this up and down, the blue area increases or decreases accordingly. And here are 3 examples. If you cut at this point here, the first motor unit, which is this one here on the outside, has this firing rate. Motor unit in between fires slower, and motor unit even higher threshold fires slower. And if you then convolute, if you add some noise, remember these are average values, by the way, so the system has noise, as I showed you in that previous slide when I filtered. If you add noise back, which happens to be Gaussian, by the way, then you get the actual firing rates of the motor units. This is the timing that you would see these things occurring if you're looking at the muscle. And if you put the force twitches in there, we know how to define these mathematically by this equation here, by beautiful work by Rakyova and Alajov. They provide us with equations that makes a set of functions like this. This is the force twitch of every motor unit. And if you put that together, those are the firing rates that I showed you. Here are 3 examples of the force twitches, and these are the forces produced by the individual motor units. The first motor unit, the lowest level one, has very low level force. Force twitch is small, produces very little force. This one here in the middle somewhere, this muscle here, the vastus lateralis, has about 600 motor units. This one here is number 190. You can see it produces higher level force, but it has some fluctuation. An even higher threshold one has a greater force twitch, produces greater force, but even more fluctuation. You're beginning to see that the higher, as you increase the excitation to the muscle, you recruit the higher level motor unit, And each one of those produces a more variable force contribution. Your neuromuscular system is set up to control accurately small level forces, where the motor units produce smooth forces. As you increase your force output, if you try to lift a 50-kilogram sack of cement, you're not concerned about accuracy. You're concerned about absolute value, and the sacrifice for that is the instability of the force we generate. But that has worked well because you've survived 4 or 5 million years, so it works. But it doesn't produce for very good control at high-level forces. So if you try to mimic this approach here in the design of robotics, you need to be careful about that. Is this what you really want to do, or should your robot be doing something else? And don't forget, your robot is not limited to that hierarchical response structure that I mentioned. Your central nervous system has to deal with that, and this is how it chose to solve the problem. Now, let me just summarize this stuff here vis-a-vis what the previous dogma was. So, we call our findings, the fine rate spectrum, the onion skin principle, because it looks like, like if you ever peel an onion, you just keep going and going, and the inside layer gets smaller and smaller. That says that small diameter motor units have high fine rates, low force twitches, and they produce low-level forces, but smooth. This is a poetic schematic, whereas the higher motor units are recruited later, produce less force, but start to have some ripple, and the higher threshold motor units are recruited later, have higher twitches, produce lower gyros forces, even though they could produce very, very high ones, but because they fire very, very slowly, they produce low forces with a great deal of ripple. And if you sum that up, you would get a force that would add up like so. But when you get to high force level, it's rough, because these motor units are firing slowly, and they're the largest ones, and they will dominate significantly the summed force that comes out of the muscle. And the muscle is not maximized to produce its force. It's working at less than its capacity, because if it worked as it was previously said to work, well, let's describe this one here first. What are the advantages of this approach? The first advantage is it enhances a combination of force, magnitude, and time duration. It appears that our system has evolved so that you can generate quite a bit of force, not maximal, but you can sustain it because you don't fatigue the high threshold motor units because they're firing slowly. And biochemically, they tend to fatigue quickly. So over time, somewhere along the way, evolution learned that, but still chose that as a solution. So what the central nervous system seems to have evolved is to produce force in a way that enhances a combination of force, still gives you quite a bit of force, and keeps going for some time. The advantage of this approach is that it serves the so-called fight and flight response. That is, if you see a tiger coming at you, you want to get away. But you want to get away for some distance. So you want to be able to sustain your force. Because if you produce maximal force at a higher rate, which was the previous concept, you would fatigue very quickly. 20 seconds, by the way, is very quickly. And chances are, the tiger would have fun. You will not go home, you will not have fun, and you will not pass on your DNA. So another approach seems to have taken over. What's another advantage? The other advantage is that it makes good use of the motor units at the beginning. They produce very smooth forces. Now, most of what you're doing right now, you're probably somewhere between 5 and 10% of your maximum force level is all the muscles in your back are using right now. You don't use high forces very much. This is where you work most of the time, the blue one. So it does a very good job at controlling the low force level, which is most of what you do in your life. And that's what I just said. So these are the advantages of the system. What are the disadvantages? The disadvantages is produces lower force than it could. This is the maximum force here, doesn't get up to that. And that's actually an important thing, because it says that you need to have larger muscles to produce the same force, which means you need more energy intake to support a larger mass. And 2 million years ago, this is important. You could run as fast as you run now with smaller muscles if they had been organized to maximize their force. But that strategy apparently did not work. And the reason it didn't work is that you fatigued too quickly. Now, the alligator has solved this problem differently. An alligator has, most of them are white fibers, which are like our high twitch fibers. And an alligator can run very fast for about 30 seconds. After that, it flops. So if you can get away from an alligator for 20 seconds, you got a good chance of living. But the alligator, when it stops, it puts its belly down, and it doesn't need to worry about being eaten by somebody else, because they have this exoskeleton that protects them dramatically. So that's their solution. We didn't develop a large exoskeleton like that. Our solution was to see if we can get a little bit further away. This is the way it was taught to all of us before. That is, the low motor units had the lower threshold motor units had lower fine rates, and they produce lower forces. The middle ones produce middle forces. And the high one, the high threshold ones, produce higher forces. The problem with this approach is it goes on for a while and then fatigues. And I've already mentioned the disadvantages of this approach. And for those of you needing to see them one more time, this is a comparison between the two. And the differences lie in that area in pink. So this is it. We've evolved this way. We've sacrificed smoothness and high force levels. We've decreased the maximum force that we can reach, but we don't do this. We don't fatigue. And that apparently must have been a critical issue along the survival path. All right. So what was the message there? The take-home message was that if you were to look at the electrical signal at a muscle and try to use the information in that electrical signal to control a robot that was activated with linear motors or DC motors, this might not be the way you want to structure your, the control. Because you're looking to do different things in the robot than the central nervous system has to do with the muscle. Because the muscle is built the way it is for many reasons, and it has to satisfy those needs. Now, if we very quickly in the next 5 minutes look at what happens when you look at more than one muscle. Everything I said up to now is for one individual muscle. What happens if we, let's look at another thing here, what we call the common drive. That is, remember I said you can think of the system as having a direct single input from the brain to the anterior horn cell, where all the cell bodies of the motor unit motor neurons are. Here's a contraction that generates a fixed amount of force. These are the fine rates of 4 motor units that we're able to see from there. If you cross-correlate these fine rates with each other, you get this. What is remarkable here, that the highest value, first of all, these fluctuations are not just accidental noise. They're actually fluctuation in the control signal, to which all the motor units respond at about the same time. There's 0 lag between these. So there's a common command that comes in from the central nervous system, which previously I described as a DC input. Turns out now that when you look at it more closely, that DC input has some fluctuation in it. But what is interesting is that all these motor units, these ones here, and many more if you look at a larger set, are controlled at the same time. That is, they respond to a common input, even if the common input has a fluctuation. Well, that's interesting. If we were to look at a lot of them, I just showed you 3 or 4 there, but here's one. Here's one from a muscle that has about 30 motor units, and the combinations are staggering. But what you see is, in all the motor units, except for a few down here, you have this cross-correlation at 0, and truly the whole muscle responds like this. Now, what if you look at pair of muscles in antagonist and in synergistic mode? Synergistic is when they act together. Antagonist is when they don't. Here is an example of the human body, one of 3 examples in the human body, where a joint, this one here, is controlled by only one muscle that does this, and one muscle that does that. And that's the extensor pollicis longus and the flexor pollicis longus. One connection goes to the top part of the phalanges, and this one here goes to the bottom part of the phalanges. If we record the signal from these 2 muscles, and there's another pair. That one here was the antagonist pair, and this one here is the synergistic pair. That is the contraction. When you do this, you're using 2 muscles here, marked in red, the extensor carpi radialis longus and the extensor carpi ulnaris. So when I do that, I'm using both of these muscles. These muscles are working together as if they were one, at least mechanically. When I do this one here is working by itself, this one is working by itself. But when I stiffen the joint, they're both working at the same time. When I stiffen the joint, they're both working at the same time. So let's look at the fine rates of these motor units in these different of these muscles in these different modalities. And what you see is, in fact, the cross-correlation exists everywhere. Even when this is the motor units from one muscle, the fine rates of motor units of one muscle are cross-correlated with the fine rates of another muscle, they still have 0 phase lag, or near 0 phase lag. That means that the command to both of these muscles is going at the same time to both muscles, when the muscles are working as one, that is, as a synergist. And I want to summarize that with this slide, that if you're looking at 2 muscles which control the joint under the condition of activity flexion, extension, and then co-activation, that is stiffening of the joint, then when you work through all the details, you can summarize it that you need 3 different control commands to achieve the control of muscles when they are acting individually, as a synergy, or as an antagonist state. And that is a signal directly to one muscle when they're working as synergist, a signal to another muscle. So this is one muscle, there's another muscle, and then at the same time, you need another signal to co-activate both muscles. These are the values of the cross correlations that are taken within one muscle, within another muscle, and across muscles. In the synergistic pair, this is what is needed to achieve that. In antagonistic pair, that is when they're opposing each other and still stiffening up, you still need all of the blue, but beyond that, you need this red that comes in, which is inhibitory from one muscle to the other muscle. When you have this structure here, the muscles can work independently in one direction or the other, or co-activated, whether they're synergist or whether they are antagonistic. Now, this is how it works in the body. If you were to do this with two motors, one against each other, you could do all of this, but you would not need that structure. So the message, again, that I want to come to is that when people speak to you about biologically inspired controls, which seems to be one of these modernistic catchphrases, please keep in mind that the system, like for example, the neuromuscular system, the control for it has evolved to deal with it in the way it is built, which is not just a simple movement, but derived from other things. For example, you may need to do a quick reflex response in the case of the kidney, whereas in robot control, there are different ways of taking care of that, but don't need non-complex control structure. You can be inspired by it, but don't copy it. At least think carefully about it. And that's the end of the message. So thank you very much.
Thank you very much. And we have time for a couple questions.
Very good. I missed which muscle you were. I can speak loudly. I missed which.
Oh, for you, okay. Thank you. I missed which muscle you were working with in the first part of the study, but do you find differences in these properties between muscles? For example, those that are used for fine control versus something that's generally just for massive force output?
Yes, a good question and right to the point, and I did not mention it. So the answer is we have looked at many muscles, including the first dorsal interosseous, which is this one here, which is almost invariably used for fine control. And the vastus lateralis, which is this one here, which has very little to do with fine control, and several in between, but I use these two as the extreme examples. The relationship of the fine rate, that is the onion skin property, is the same. That is, even these two muscles that are essentially extreme in terms of how they are constructed, they are controlled the same way. What differs is the degree of the fine rate. They either, the first dorsal interosseous muscle as a unit all fires faster. The vastus lateralis as a unit all fires slower. And the second thing, which I did not mention at all, is the distribution of the motor unit recruitments throughout the muscle. In this muscle over here, the first dorsal interosseous, most of the motor units are recruited at the beginning of the force level. So that the incremental increase of every motor unit is small on top of the other one, and the fine rate is also greater. It makes for smooth force transition when you recruit a new motor unit. So you don't see this sort of stuff, but rather so. Because it only has 120 motor units, that muscle, the vastus lateralis, it has 600. So let's assume, this is not correct, but simple assumption, that each motor unit has a unit quanta of force that it adds. If you have 600, every time you recruit one motor unit, it's smooth. So it doesn't have to worry about that. And as a consequence, the fine rates are lower, which means it doesn't have to smoothen out the force of each one of the motor units. So there are these distinctions among muscles, but the main concept, the spectrum of the fine rate, is identical. Simple amplitude parameters vary, and the distribution of the motor units within the spectrum varies. They begin more at the beginning and the FDI, whereas the vastus lateralis, they're spread out throughout the full range. of the force. But we looked at maybe 24 other muscles in between, and it's the same.
As far as I understood, all the examples you gave were for isometric contractions. So does the same thing hold true for concentric, eccentric contractions?
For eccentric contractions, I do not know. And I will tell you why, and this is going to be reproduced. I'm going to be very careful with my choice of words now. So the answer is yes. All the data I've shown you was for constant force, not constant force, for isometric contractions. The reason for that, it makes the technique much easier. Things don't change very much. The signal is relatively stable. Signal processing, certain kinds of signal processing techniques can be used under those conditions. So the answer is yes. With respect to a dynamic contraction, which of course is what we really want to study, let me say the following. It is not impossible to do it. It is very difficult to do it. And someday soon, you're going to see somebody do it. With respect to what I expect from that, I do not expect to see much of a difference with what happens during constant force contractions. But of course, what differs, motors are going to go in and out faster. So you can see how technically that becomes much, much a bigger challenge. But I think the recruitment order, the firing rate spectrum, I strongly, strongly suspect will remain the same. It will be dynamic though. That the operating point that I mentioned to you, that's going to go like this, instead of just progressively up and down. But the image it's going to traverse will remain the same. That is a hypothesis, but I'll bet you 25 cents on that.
That was a wonderful talk. Thank you very much. I actually have two questions. One is just to clarify something. The distinction between the situation with Eccles and yours was, I think you said largely because they were stimulating the motor neuron directly in the spinal and you were getting the brain to actually activate it. Is that right?
Precisely.
The other question that I had...
If I may add on that before you cross, I want to be clear here. Eccles' data is correct.
Right.
It's just that he was electrically stimulated.
Whereas you're looking at natural recruitment from the...
Yeah, but I think that our motor units can respond the way Eccles did.
Of course.
If we were to electrically stimulate the high threshold motor units, they will fire faster. It's just that the system does not allow that. And whereas under electrical stimulation, it's like a race car driver. You know, you have a great race car, but you're not necessarily going to win a car race because you have a great race car. You need a driver in there, and the driver controls that car to win.
And this sort of gets at my second question a little bit. I work on insects, and of course, the arthropod situation is very different from what you're describing.
Which situation? I didn't hear.
With insects, arthropods?
Yes.
Where you have a very small motor unit. You only have one or two motor neurons going to each muscle. Yeah. But frequency code becomes more important. There's a bunch of integration that's going on there. So the way the brain, or the thoracic ganglion in this case, is controlling those things is a little bit different. I was wondering if you thought about how those two situations differ.
So I have no experience and I mean, I've read things as you have mentioned. But what I can tell you the following, I'm going to bring it back to the human picture. If you look at the external oculi muscles, these muscles here that control the movement of the eye, they're very small. They fire very fast. They go up to 70, 80 pulses per second, which our limb muscles do not do. And there's only very few motor units within each one of those muscles. 10, 20, and so. So you're approaching, I don't know how close to an insect that happens, but if you look at these muscles, this holds. Again, it scales up dramatically. Instead of the high fine rate being 20 pulses per 30 pulses per second, so you may reach 70 or 80. But the first motor unit fires the fastest, the last motor unit fires the slowest.
So that's very interesting. It may be that the difference is not a difference between arthropods and vertebrates, but small and large muscles, because the arthropod muscle doesn't spike, and so it uses the neuron to conduct up and down, and so it may be that if you get really large muscles, that just won't work.
It doesn't happen.
But they're both converging in the same force.
Yes, no, I understand, and I tend to agree. But remember, in a large muscle, you don't have to do that. But in a small muscle, of which this is the best example I can think of right now, at least the one with which I have experience, you're getting close to what you're seeing.
It's a short question. I'm not sure whether you answered it already. I don't hear very well. What consequences does it have for electrode stimulations if you do muscle experiments?
Yeah.
So hooking a nerve up or EMS, the different techniques, what consequence does it have?
Yeah, so, yes, this is actually a good question. So if you're going to electrically stimulate, say, a reasonably large muscle for the purpose of rehabilitation or something like this, now you need to know that when you, excuse me for a second. Generally, the way electrical stimulation is done, at least in humans, is that you place one electrode either on top of the muscle directly or near a nerve source. It's better to do it on top of the muscle, it's less painful. You're stimulating all the motor units, that is the muscle, you're actually stimulating the branch of the nerve that goes to the muscle fibers of the motor unit. They're all responding at the same fine rate. Most stimulators that I know so far, you put pulses in there at 20, 30 pulses per second. So what happens all at once, not in some hierarchical order, all at once, you're making the low threshold motor units fire 20 pulses per second, or 30 pulses per second, and the high threshold motor units fire the same rate. And if you did that, the tiger would get you. That is, you fatigue, which is a common problem with electrical stimulation in rehabilitation environments. And it does so because of the electromechanical characteristics of muscle fibers, which are relatively well understood. So, it gets very tricky here. It should be possible to solve this problem, but it's not easy, because you have to know, you have to be able to change the fine rate of a motor unit that responds before the other ones do. So you have to have some stimulator that adapts the fine rate. That's a very difficult thing to do. And the problem, this makes even more evident the issue that has been observed for decades in clinical environments, that when you electrically stimulate a muscle, you tend to fatigue the muscle quite a bit. Now, there was work done in the 80s and the 90s where they showed you can change the fiber typing due to excitation. But that didn't prove to be as positive as we wanted. There's an issue, you have to maintain the fiber typing in an altered way that becomes a problem. So I'm not giving you a very good answer, but I don't know what a good answer is.
Experiments. If you look at muscle properties, if you measure muscle properties in animal experiments, you do also, you look at the firing rate, you turn it up, the firing rate, and you look at the proper amplitude where you get a maximum amplitude, but it's probably a very rough compromise which you get at the end.
Yeah, it's good to learn about the individual properties of the motor neurons. It's a wonderful way of studying them. But it's completely obliterating the notion that there's a control system behind the muscle. That's how you and I, that's why you're sitting down and not falling down on the ground right now, by the way. So what our data shows, I think, a little more than previous work shows is how, in fact, the muscle is controlled and the organized, structured way in which it responds. And when you do these electrical stimulation experiments, you put aside the whole business of control.
I think we should stop the discussion now because we are running into the coffee break. I thank you all for the interesting question. I thank Carlo for the presentation very much.